thanks. And once again, we're going to be talking about gut and immune health. And really, I'm going to be focusing primarily on the lower GI and how the microbiome that inhabits this region really intertwines with the overall immune system and immune health in the body. And one of the things that I really like to do anytime I'm going into one of these subjects and subject areas is try to look back at some of the landmark historical studies that have been done. And as I was prepping for this one and looking into a few of the more classical trials, I came across what I thought was one of the most interesting and just blatantly elegant studies that I've ever seen. And what it was a study by Collins and Carter in 1978, where they really proved that stable microbes in the GI help animals and help us resist infections, particularly ones that target the GI tract. And for this, they used a germ-free animal model where they took these mice, and the mice were raised in an environment that was devoid of any kind of microbes. So what we know is that when you're born, most of the microbes that you're exposed to are the ones that colonize your body. And being born into one of these germ-free environments can actually limit any kind of a microbial cultivation in the GI. And then what they did is they took that group and they compared it with a conventionally raised animal group. So some mice that were just raised normally that had a normal developed microbiome. And they infected each of these groups with various doses of a salmonella pathogen. And what they saw in the germ-free group is that it took only 10 cells of this salmonella pathogen to induce a lethal infection. And compare that to what they saw in the group of conventional mice that required 1 million of these salmonella cells to induce a lethal infection for them. So what that really keyed in was just how important these microbes are to that particular defense from this pathogen. And in addition to that, they went further and they looked at just, okay, so is it that microbial direct interaction that is causing this? So they also took a similar group of mice raised the same way, and they injected them both intravenously and from an intraperitoneal injection under the skin. And they saw that both these conventional and the conventionally raised and the germ-free mice actually succumbed to a similar dose under this. So it really came down to that oral effect. But beyond this, some of the new trials that have been done, they've looked at how this germ-free group these germ-free mice, how their GIs develop and what about their GIs is different and how that impacts the immune system. So we know that it impairs the overall GI development when there's no microbes. They have smaller pyre patches. They have fewer CD8 and lymphocytes. They have lower levels of mucosal IgA antibodies that are produced. And overall, they do have a pulmonary susceptibility that's also been outlined. And when we look at the GI and how it connects to the immune system, the GI can actually be thought of as the largest immune organ in the body because of how many immune cells are found in that region. I've seen numbers that tout it to be over 70% of our body's immune system being located in the GI. And what this does is it's really good for both the immediate GI specific portions, but also our GI is kind of our whole body's sewage system, right? So we have all the food that we eat that goes through us, and we take what we need from a nutrient standpoint. But it's also one of our main drainage pathways that we have in the body. So anything that's any surface on our bodies that can really that any orifice really, it can wind up draining a certain amount into the GI through the throat. So what that allows is the body's GI tract to actually get exposed to all sorts of things that our body can be exposed to. And this is really important because of all of the different microbes there, in addition to all the immune cells that are housed in the GI tract. See, these microbes that make up this external environment population that lives within our body, they are really important from a number of different factors. In the previous presentation, it was talked about how they metabolize all sorts of different plant materials and how those interact with the microbes in order to induce beneficial effects for us. But these microbes themselves will interact with one another, and they fight off plenty of infections. As demonstrated in that classical study, they can be really good at this and really efficient. But there's also just a general priming that happens from all these microbes. So it's really hard for our immune cells, which are greatly outnumbered by the number of microbes in our body, to know which one of these microbes that's in there is a friend or a foe. So they really do a good job of working with the resident beneficial microbes that are there, the commensal species that it's used to having, to work together and signal when there's something that would be problematic. So we haven't fully outlined all the different ways that the microbes that inhabit our GI, how they interact with our immune system and how they directly signal everything. But we do know that it's this symbiotic relationship as a general total group. And therefore, it's really important that this grouping is cared for properly and we really tend to it from a nutritional standpoint. In our GI, we have an upper GI and a lower GI. And throughout this, it's a changing environment. It is very different in the different regions, with the upper GI being where we have so much of that early breakdown of food. This happens in the mouth from the saliva, as well, all the way down to the stomach, where you have this incredibly low pH that's really important for sterilizing a lot of the external species that do make their way through. So that way we're not constantly facing the greatest, strongest possible challenges at all times. And then as you move into the lower GI, you have more of a neutral to slightly acidic pH here. And that helps control some of the growth of a lot of microbes. And as you move through the GI, you do see increases in the amounts of the microbes that are present, as well as the diversity of the different microbes that are present. So you look in the stomach, you have a relatively low diversity of microbes because really not many can survive in that region due to that low pH. So usually the ones that are there, they're very hyperacid tolerant for a number of different reasons. But some can bypass that region and some of the things that we eat. So we can see things like when we consume a probiotic, they have a certain level of acid tolerance, but that doesn't mean that they're going to be able to survive constantly in the stomach. As you move through the small intestine, the further you get towards the colon, the diversity increases. But ultimately in the colon, it's just such a major factor higher than the rest of the other regions of the GI in the sheer magnitude of cells, microbial cells that live in that region. Then you have diversity differences that are also in the architecture of the GI mucosa. So the ones that live on the epithelial surface aren't necessarily the same as the ones that are floating around in the intestinal lumen that call that space home. So you have differences there as well. Our microbiome, it develops from birth. As shown in those germ-free mice studies, it's a very important thing because of the environmental influence. So now with the advent of all this next generation sequencing and about probably a little bit over 10, maybe 15 years of research now, where we really are diving into the microbiome. So that would be all the collective genetics of all the different microbes that are in this region. And those genetics, because there's an increased amount of diversity in these populations, those genetics far outweigh the genetic diversity that we see within the human genome. And some of the factors that the environmental factors and relatively uncontrollable factors, as well as the more controllable ones that impact our microbiome, they start in humans with the birthing process. We've seen big outlines in the differences for cesarean delivered babies versus vaginally delivered babies in the initial inoculum that winds up setting an infant in one direction or another. That pairs with the infant feeding method, where we see differences between breastfed and formula-fed infants. And then within formula-fed infants, very specific formulas can induce different microbiome influences. And as you grow older, the life cycle stages that change, those wind up having associations with differing microbiomes. So as we age, usually what we see is our microbiome starts to diversify more and more, but different stages of life wind up seeing different microbiome profiles overall. And then we have something like geography. And I think the effect that geography plays in the microbiome might be the main reason it's such a difficult area to study and draw grand conclusions from. Because a healthy microbiome as a baseline, knowing what that is would be just the absolute goal of microbiome study. But because of geographic differences, we see that microbiomes, a healthy microbiome in one region of the world is drastically different from a healthy microbiome in another region of the world. They both are able to serve beneficial functions to a person, but they look very different. So in that sense, it becomes very hard to say, oh, one type of microbiome is healthy. Other things we know that impact the microbiome, stresses, physiological or psychological stresses, or even things that are beneficial good stresses, stuff like exercise. Those can impact our microbiome. And that's one of the more controllable features that we have. As well, back to geography, regions that you're in. So whether you live in a rural or an urban environment, or your ability to move and change your environment, that can greatly impact your microbiome development. Then you have things like pharmaceutical treatments. Antibiotics are the most well understood of these, since they are designed for their ability to kill bacteria. Those bacteria aren't always going to be the target of the pharmaceutical intervention. So there's a little bit of a friendly fire, you would say, for that, and that can really impact how a microbiome changes. Other pharmaceuticals really can impact this as well. One of the major ones that's been identified to really be able to impact the microbiome is something like proton pump inhibitors, but also things like pain medication that can really impact a microbiome. And then our I would say probably the most controllable of these microbiome influencers would be an individual's diet. And when we look at the diet that we see, there's all kinds of wish lists for things that we would want to see people consume. But more often than not, in the United States, we're fighting with diets that can look pretty bad. And these are usually going to be diets that they're just due to our modern society and modern convenience and advances. We've seen that energy-dense foods that have the ability to deliver a lot of calories, taste very great, taste almost, I mean, there's a reason people continue to consume these. They're convenient, they taste good, and they're widely available. I made this presentation right around the time that the Super Bowl was going on. So one of the things that I thought, even during a more of a socially distant Super Bowl, that was interesting was just how much I still fell into the pattern of not eating great during the Super Bowl. So it's just a little microcosm of how problematic the diet can be. Because we know the diet does play a role in both GI immunity as well as influencing the microbiome and chronic inflammatory effects on the diet. So there are certain dietary stimuli that have been outlined to directly impact our either our immune defense via response from certain infections or chronic disease development, as well as things like systemic inflammation and allergy autoimmunity. Certain things can elevate inflammatory cytokines. They can target things like toll-like receptors. In this graphic here, I have a lot of arrows and a lot of words, but what you can see here, the authors from this publication, they really outlined a nice pathway towards things that impact the GI and GI inflammatory states. As well, they looked at a few things that have more of a positive effect. So that would be signaled here by those red lines with the blunt ends. Some things are a little bit less outlined as being positives or negatives. Those are outlined in the dotted lines that are going towards the next thing. So what we see here is that certain dietary stimuli have negative effects and positive effects, and those really tie together through dysbiosis as well. So you have things that can directly impact our GI and directly impact the microbes that live there. And back to that microbial population. And so what makes it up? What makes up our microbial population in our GI? I mentioned dysbiosis earlier. Understanding where we categorize these microbes as either potentially good or potentially bad or a little bit in between can become important. So the terms that are used here would be commensals, symbionts, pathobionts. So the commensals are those permanent residents that are in our bodies, and they provide a much needed general diversity of microbes. They also can pair together with other microbes to make these little collective groups that work together to do things. But the ones that have been outlined as having some sort of a beneficial function to us, be it they make a metabolite that is helpful for us, or they have some direct effect on the immune development or the immune system, they wind up impacting the environment around them, or they've just been associated with some sort of a health benefit where you know that something like FPROW, so fecal Bacterium Prisnitzii, I definitely butchered that one, but we know that that has an association with certain digestive conditions where its absence is higher or has been more associated with incidence of those conditions. And then, contrary to the symbionts, you have the pathobionts. So these would be also resident microbes that are present across many microbiomes, but when they are at amounts or they can cause issues when they reach levels and proportions beyond that normal amount, where they're no longer being well controlled by the other groups of commensals and symbionts in the body, or they wind up in a different spot in the body that can start to cause issue. I also have highlighted here something called the serious disruptors, as I like to refer to them. And these would be a lot of your classical microbial pathogens that enter the GI and cause issue. So viruses, rotavirus, things like salmonella, enterotoxigenic E. coli, as well as something like Campylobacter. Just as a general group, what these are things that we acquire environmentally, and they may not be long-term colonizers, but they can introduce very dramatic effects in a short period of time and drastically change the microbiome. And the opposite of these that you would think about is actually probiotics. So probiotics are going to be these environmentally acquired microbes that we take for their specific benefits to us, but they're not necessarily going to colonize an individual permanently. And it's all about helping the body maintain balance to be able to fight off all sorts of issues. So for each individual, you have what's called a homeostatic state. So homeostasis is where you, for a healthy individual, would be when you have a microbiota that has a high number of diversity in both the number of different types of species that are there, as well as the function of these different species. So you want to see overlap in all these functions. So that way, as our body responds to all sorts of stimuli, certain microbes increase when others may decrease. There's overlap between the right functions that we want them to play. Also, this provides an ability for an individual's microbiome to resist change that happens under physiological stress. It might bend a little bit, but it's not going to break fully into a whole new environment that can be incredibly problematic. When there is an underlying issue where an individual has a homeostatic state that is problematic, or they're moved into a transient state that is more problematic, this term is often called dysbiosis. And this is where you see things like an expansion of pathobionts, a lower level of total species diversity, or fewer beneficial microbes. And it can be each one of these individually or a collection of all of them kind of mixed together for individuals with dysbiosis. Oftentimes, this dysbiosis is also paired with some functional issues for individuals. In the question and answer session earlier, we were talking about looking at things like intestinal hyperpermeability or this leaky gut syndrome. Something like that is more of a functional thing that can be paired with something like dysbiosis or the presence of certain conditions or symptoms for individuals. The microbes on the GI, they make up one of these barriers in the layers between the GI and the external environment in our body. And there's more layers than just the microbial layer. You have a chemical barrier made up of secretions, a physical barrier of all these epithelial cells, and then an immunological barrier. And the makeup of the chemical barrier, it consists of a lot of secretions like secretory IgA, but the primary component of it is this mucus, so mucin, that coats the epithelial cells, and it's a region where certain microbes can live, and it helps lubricate and prevent a lot of the damage from damage in the physical barrier from a lot of the external components, including those resident microbes. So it's a nice little buffer zone that our body creates, and it's primarily made up of this glycopeptide where you have a protein core in the mucin with oligosaccharide side chains. And I really want to point out here the terminal sugars on these mucins. So these are the oligosaccharide side chains coming off of that protein core. One of the monosaccharides that's very present across the mucus is what's called fucose. And a fucose monosaccharide, it's relatively rare in the diet to see, but it's something that does exist in this mucin. You also have sialic acid as one of the other main components here. And certain microbes, only a very select group, but a few of them are able to either bind to this to initiate their colonization, or some others are able to consume mucus as an overall food source for themselves. The tight junctions that make up the physical barrier that connect each of these epithelial cells, the goblet cells, and the other cells that are in this epithelial monolayer together and help seal that barrier from the outside world. They contain these tight junction proteins, and these tight junctions are really fantastic when it comes to preventing large molecules through and forcing things that would be larger to have to be taken up transcellularly instead of just being able to leak through the small little regions. You do get some uptake of very small things like certain minerals, but these are really good at preventing a lot of the microbial components as well as larger food components that could be problematic. Once again, there are all these immune cells that do sit below this layer that are able to respond to all sorts of events and be recruited by the body to different regions to deal with other events that happen. There is a connection between our body's immune system overall and the GI. I mentioned earlier it's that sewage system. It samples it constantly, and it's always being prepared for all sorts of things. And this is why that mucus layer, it does need to separate that component, those components very tightly from anything that is being introduced to it through the diet or through any kind of drainage secretion that happens. That compartmentalization, it is a good way that the majority of that immune system is limiting exposure of these commensal microbes or even the problematic ones to help not have our bodies go through an excess immune response constantly. And what we know also is that this is part of how our body does connect the systemic immunity as well as the microbiome together. So we have this we have these various axes that have been outlined in recent years. You'll hear often the gut-brain axis, the gut-liver axis. There's also something called the gut-lung axis, where certain events that happen in our GI and are all mediated through these microbial interactions, they start to have downstream effects within the body. This is an area of expanding research constantly, and it's one of those things that I think the more of the less conventional medical bodies have been really privy to for a very long time, about just how important gut health is and how important the food we eat plays a role in whole body health. How that is now starting to finally be caught up to in the scientific base as well moving into more of a clinical understanding that we have for it. So gut permeability is an area of that we can really look at and focus on when it comes to the immune health in the body. Certain conditions have been associated with this intestinal hyperpermeability. You have certain autoimmune conditions, something like Crohn's or ulcerative colitis, type 1 diabetes. This article here, they were able to look at the incidence and the percentage of patients that had a certain condition that also had a paired intestinal permeability. So not all of them had it, but these were the conditions that did have a high amount of correlation between intestinal permeability and presence of those conditions. So certain ones show up a lot with a joined intestinal permeability. Something like a chronic liver disease with type 2 diabetes was found to have intestinal hyperpermeability in about 65% of those patients. And connecting the permeability condition or permeability issues with the microbiome population can actually influence this directly. So when you have dysbiosis, either depleted total diversity, depleted symbionts, or colonization of these excess problematic species, oftentimes what you see is that there's a depleted level of these chemical barrier secretions. Maybe certain groupings of these microbes are really efficient at consuming mucin. The individual has a poor diet, so they're not introducing prebiotics or other beneficial fermentable units into the diet. So all of a sudden, this GI is very at risk of breaking down fast. And then what happens is the immune system is having to respond to these threats. And what happens then is you have sort of this snowball cascade event where gaps in the tight junctions allow for more epithelial translocation and more microbial subunits. Even oftentimes when these things aren't alive, they're broken down, they can still elicit immune responses. Those immune responses, as Jen was pointing out in her presentation, have the ability to have a very violent reaction with a lot of reactive oxygen species being produced, and that can damage the surrounding area, leading to more problems and more responses being required. So looking at ways that we can quench some of this with dietary approaches, as well target that microbiota so you can really help the body heal from this, is going to be very important for maintaining health and maintaining a proper immune response for the body. Identifying potential stimuli and stressors. You have certain things like antibiotics, diets. These all can affect the intestinal microbiota. Also, those prescription drugs, travel, gastrointestinal stress, and lifestyle. They can put you in this altered intestinal microbiota space, and it might not be permanent, but it can lead to compromised immune defense and inflammation in the short term that can turn into more of a long-term issue, in addition to that hyperpermeability as well, something like metabolic dysfunction. The stability of the microbiome is really important. So helping an individual have more of a healthy, stable state is really important. You want that to be able to resist colonization and respond anytime there's all kinds of these stresses. So you want a species-rich, healthy, stable state. But what can happen is from disturbances that can move an individual into that degraded transient state of dysbiosis. And if that moves far enough in one direction and the recovery becomes not possible, then what the problem is, individuals move into a stable degraded state that retains a lot of those same properties that the healthy stable state has with its resistance, but that in turn is now resisting beneficial effects. So this is why working on the preventative end is very helpful, but recognizing this and taking it into account as interventions are done is really important. So dietary approaches are very helpful, and we see in the research now that dietary influences in the microbiome are very strong and can be very vast in how they affect the microbiota. The best data here so far is around the Western versus something like the Mediterranean diet. So these are just general diets. One, you would say, oh, that's a bad diet, in the Western diet. And the Mediterranean diet, it's full of high levels of antioxidants, fiber, and really high quality, good proteins that individuals are getting. So you see in the Western style diet, decreased total bacteria from a species standpoint, from a diversity standpoint, decreased beneficial species in bifidobacteria and lactobacilli, but also that's going to be contrary to what you see in a more of a beneficial diet, where you have in the Mediterranean style diet, where you have increases in the bacterial diversity and increases in all of these beneficial species to encompass a lot of your diversity. Other things to note, certain diets are very beneficial for treatments for individuals. Something like the gluten-free diet to help control an individual's symptoms is a very helpful approach. But by moving to that, a lot of components in the diet are lost that can drive a lot of the microbial diversity in the body. So when an individual is on a specialty diet, trying to make sure that they are getting enough of these microbiome effectors. When we look at things that can impact the microbial populations in the diet, we often think of prebiotics, but there's also non-prebiotic components that can affect the diet. So probiotics, proteins and fats, vitamins, antibiotics. The prebiotics, you have your classical ones like your oligosaccharides, fructooligosaccharides, and galactoligosaccharides. The fructooligosaccharides would most people would recognize inulin as being a long-chain fructooligosacchαρide. There's also plant phenolics that can impact the microbiome. And I like to call these phytobiotics because they have two ways that they really seem to do this. They can increase the growth of certain beneficial species, but they can also help control the growth of problematic species. And then you have some more novel prebiotics like a human milk oligosaccharide, and I'll touch on that one in a little bit. Probiotics are those tourists that you can throw into the GI economy, and they can elicit a lot of these beneficial effects for individuals. And for probiotics as a general group, some of them have some really well-shared general effects that they have with each other, where they can contribute to colonization resistance from outside sources. They can normalize certain microbes, produce things like short-chain fatty acid, have pH benefits, regulate intestinal transit, increase turnover of enterocytes in a good way. As well, they have things that would be more of these species-level effects that aren't shared by each probiotic that's just classed as a probiotic, but you start getting a little bit more specific in what they do. And then ultimately you have a lot of these strain-specific, really rare effects where you start seeing things like very specific immunological effects where probiotics are being delivered for that benefit to individuals. Prebiotics, on the other hand, are more of a general food and things that are given to these microbes. And these can play a role in our immune defense as well, but they feed the general population. They're not going to just be one specific type of microbe. There's going to be prebiotics that can be very specific in what they feed and feeding a large general group of good microbes, but also feed a lot of the commensal species and just the general growth of microbes in the GI. Through prebiotics, you can get the growth of a certain beneficial species increased, and that can increase the amount of antimicrobial substances that those beneficial species produce. As well, you can also see things like fermentation end products, certain things like short chain fatty acids that can help decrease that luminal pH a little bit, and both of those help to control pathogen levels. And then you have something together that these can do with this altered overall microbial profile, and with prebiotics, it's probably the best way that you can look for more long-standing targets of something like the microbiome population in an individual. With specific impact on the immune system and immune effects, this trial here is a meta-analysis that coupled together both pro and prebiotic interventions and looking at how they target the immune system once they're delivered. And the measure here in the forest plot is the presence of C-reactive protein in circulation. And what this study was able to find was a very significant effect overall from pre and probiotics on the reduction of C-reactive protein, which is a good biomarker for the presence of excess inflammation in the body. So it's a really good way for us to be able to control or utilize probiotics and prebiotics to help control some of that excess inflammation response. Those phytochemicals from plants. Luckily, we just had a whole presentation on this right before this, but we know that these phytochemicals from plants, they can positively interact with the gut microbiota, and the gut microbiota is really important for helping to break down and modify these metabolites for us to be able to break them down, absorb them, use them for some of their benefits. As well, they can contribute to direct effects on the local microbes there. And a lot of them have been shown to drive the growth of certain species that we associate with benefits, lactobacilli, bifidobacterium, Akkermansia, and F. prau. And this can occur with these metabolites from plants. They can positively shape the microbiota. They can help inhibit some of the production of endotoxins, as well increase short chain fatty acid production. And that short chain fatty acid production, when that can be very beneficial from an overall immune response in the body. As well, you're going to have a lot of help and control on certain responses from various stimuli that these heavy antioxidant compounds that do reach the lower GI can be very helpful to have floating around nearby when that happens. Fermentation end products are another this pairs with butyrate, but these on a class in their own, they can be targeted as delivery mechanisms. So certain things like trying to increase the levels of butyrate, that can happen from prebiotic sources, introducing probiotics that we know do this, as well just delivering them on their own. So you have things like butyrate, propionate, acetate, and these are things that can be measured in some clinical assays for individuals that have levels of them. When you combine certain probiotics and prebiotics together into a form that are synergistically helping an individual, this is where you would refer to something as a symbiotic. So certain microbes certain probiotics have been paired very regularly with these prebiotics, and they can build off of one another and increase their function. For this particular grouping that I have outlined here, these are a lot of microbes that have been outlined from a species-specific standpoint for both their effects on a general health and well-being, bowel movement effects, but also their ability to support the body's immune system response. And the one in particular I wanted to highlight was the Lactobacillus casei 431. Oh, it looks like I can only go one direction on this. Hold on, I lost my control. Okay, looks like it's back here. One of the really exciting areas that I wanted to really spotlight very quickly here would be the novel potential prebiotics that are on the market. One of these new groups of prebiotics is called human milk oligosaccharides. And human milk oligosaccharides have been outlined as incredibly important components of an infant's diet coming from human breast milk. They really are a prebiotic that's been designed by nature specifically for humans. It's in breast milk primarily, but we've also been able to recently synthesize this and produce it via a bacterial fermentation process. And HMOs, why they're so unique is they have both an immune effect and a very targeted prebiotic effect to beneficial microbes. But in infants, what's also really amazing is how these act as almost an extension of the mother's immune system given to the infants, because those infants, they have a very underdeveloped overall immune system. It's still learning what things are, being exposed constantly to new microbes, anything that it's a new food that it's eating. These HMOs are really important for helping the body develop its immune system, turn over, change it, mature it into more of a overall immune system ready for all the different things that it's going to face. And it also really helps with something that's called a decoy effect because of the structures here on these human milk oligosaccharides. So here I have 4 different human milk oligosaccharides that are outlined in the little structures there. The first one in the top left corner is something called 2FL. So that's 2-fucosilactose. And you look at the structure of this, that red triangle that's coming off of it, that's actually the same fucose molecule that I pointed out when I was talking about the mucosal terminal sugar. And why this is important is those terminal sugars, they're in the mucin, and they're also on the surfaces of intestinal cells themselves. So what happens is when the infants are consuming this HMO on a regular basis, it helps to bind and decoy bind these different microbes that can elicit a lot of these problematic GI specific events. But in infants, they don't have a full immune or a full gut barrier that fully prevents these from slipping through as well. So you have certain amounts of these prebiotic human milk oligosaccharides that make their way into circulation too. So it has this full systemic effect on the infant's immune system where it does support better overall outcomes. And that's part of the main mechanism where we believe that infants, they have just better recovery as well as better prevention of certain things like diarrhea-inducing bacterial and other viral infections. So 2FL really does contain this ability to influence the immune system. And it can do it through a number of ways. This study here was actually outlined in adult animals. And what it looked at was when the immune system of an animal is challenged in 2 different ways. They looked at it from a challenge with just a simulated colitis model where the animals are predisposed to developing an inflammatory bowel disease. And what they saw was feeding about the equivalent in humans of about a 3 gram per day dosage of 2FL. They saw that there was a specific effect on inducing and increasing the presence of probiotic or of beneficial and symbiotic and commensal microbes. They had an overall increased barrier function with more tight junction proteins and less tight junction damage once this GI was challenged, as well an increase in the production of mucus and the mucus layer, but only during GI challenges, as well a modulated immune response. So it didn't have that overarching excess immune response. And this study also showed that that the animals themselves, they just had fewer side effects from these problematic stimuli. And they did it with that colitis model, but it was also repeated with an enterotoxigenic E. coli infection model in the animals. So it really is an exciting area of how something like one of these human milk oligosaccharides, and in particular this 2FL, can really support the immune system. When we think about as a targeted prebiotic, 2FL is really exciting because it has this very specific ability to resist metabolization from most microbial interactions that it has. Most microbes don't possess the ability to break this down in the way that we see with other prebiotic components. So other prebiotic components in this this table, I outlined galacto oligosaccharide. It's very good at feeding a large swath of beneficial bacteria, but it's also able to feed a lot of the more problematic species. So you have certain things like Clostridium, certain E. coli's, and things that can consume and live off of this galacto oligosaccharide. Versus what you see from 2FL is that metabolizing ability just isn't there for most of those microbes, and it really requires only a few small or only a small number of commensal species that really are able to break this down and use it for energy in the same way. So here, Bifidobacterium longum, the subspecies infantis, that's primarily in infants, but Bifidobacterium bifidum from both adults and in infants has been outlined as being able to initiate this breakdown. So you just have a more targeted delivery of a prebiotic with something like 2FL. And that doesn't mean that it's the only option that you should be using. Other prebiotics are good, but for certain individuals where they might want something that is a little bit more targeted, that isn't quite like throwing gasoline onto the fire, where you have a well-known amount of non-helpful species, that can be a more preferential choice. This graphic here is detailing a little bit more of that decoy binding mechanism, where you have these surface bound sugars and also the sugars that are terminal in the mucus, where microbes from the environment that can initiate a lot of these colonizations and lead to deleterious effects on the microbiome and the individual themselves, those can mistake these 2FL components for their binding sites, and it makes them unavailable to bind to these cells. So that's one of the main theories as to why this has such a good support for those infant immune systems. And you do see that in infants that are formula fed, when you give them this 2FL in that formula, they have inflammatory cytokines production that models more of the breastfed infants, as well as just more better overall outcomes. Additionally, these can wind up helping support the removal of a lot of the bacterial exotoxins. They can serve as ligands for those exotoxins and support their removal as well. So now, this is really important in infants. We're really learning more and more about that every day, and all sorts of studies are coming out really detailing how beneficial this is for infants. But there's also some landmark studies that came out in adults. So go back 20 or 10, 15 years ago, thinking about having enough 2FL to feed an infant, let alone an adult, that was just a non-starter and something that seemed fully impossible because you really can't find these human milk oligosaccharides anywhere else. And what we've seen now that we can produce it via fermentation procedures is that we've been able to study it in adults and look at how it impacts a healthy gut microbiome. Or how it impacts and supports a healthy gut microbiome. And what we've seen in adult trials, where adults were fed 5 grams of 2FL over the course of 2 weeks, is a targeted increase in the proportion of Actinobacterium. And Actinobacterium is the phylum, so an overall group that encompasses many of the beneficial bacteria, including all of the Bifidobacterium. And it's been outlined as a group that's really important for overall microbiome or microbial homeostasis. As well, those Bifidobacteria consistently show beneficial immune system effects. So it's really good to be able to pair all these different tools that we have together, these different dietary tools that can target things like the microbiome, helping to support a healthy individual's microbiome on a regular basis. But also, it's important to be able to target somebody who does have a dysbiosis. And targeting these dysbioses, one of the approaches that can be used in a clinic is a weed, feed, and seed program, where you pair together things like probiotics, phytobiotics, so those plant materials or medicinal herbs that have this beneficial controlling effect on the growth of certain problematic species, pairing that with prebiotics that we know feed beneficial microbes as well.